Hi everyone, I am CatarinaBranco and I am here to present the work I am currently finishing entitled Influence of the Sea Level Variation on the Genetic Diversity of Current Human Populations from Southeast Asia. It was held in the University of Vigo during my PhD and was supervised by Miguel Arenas. The environmental fluctuations have since always affected the world and changed the available resources and habitats. Given this, they presumably influenced the expansion of early modern humans. Particularly, the last glacial period, year after LGP, was marked by large changes as the extension of ice sheets, the availability of water, the sea level, and the vegetation. We would like to note that these environmental changes are related. For example, the extended ice sheets induced a lowering in the sea level since a large amount of water was stored in the ice caps. Okay, but how did exactly the LGP may have influenced early modern humans? The adverse environment decreased the available land, what caused a range contraction in the settled populations, and may have caused a decrease in the genetic diversity. Also, the adverse environment may have caused a habitat fragmentation that may have led to a differentiation between subpopulations. The negative impact that the environmental fluctuations had on the genetic diversity of modern humans have been widely studied. However, the lowering of the sea level during the LGP approached and connected distant islands, what may have favored gene flow and contributed to the genetic diversity. Here, we focus on Southeast Asia because the region comprises thousands of islands that may have been connected by land bridges during the LGP as a consequence of the sea level lowering. This may have influenced the gene flow in the region. Besides, during the moments of low sea level, the Sunda Shelf was exposed, this region here. The temporarily exposed land during the LGP may have provided additional resources for human populations. Still, some islands remain isolated, and migration through long-distance dispersal events, hereafter LDD, involving sea crossings may have occurred. Here, we intend to understand whether the sea level variation influenced the genetic diversity of current Southeast Asian populations, and if so, if it affected the demographic and genetic parameters. Our observed dataset comprised 720 samples of the hypervariable region of mitochondrial DNA belonging to 25 populations distributed along Southeast Asia. In the map you can see those populations. To perform future analysis, we classified the population into 5 groups according to their geography. Here, Group 1, Group 2, Group 3, Group 4, and Group 5. We started by analyzing the genetic relationship within our samples by reconstruct a phylogenetic tree. To infer the phylogenetic tree, we firstly identified the proper substitution model of DNA evolution, and then infer the maximum likelihood phylogenetic tree. Then, to understand whether the sea level variation induced by the LGP together with LDD events influenced the current genetic diversity of Southeast Asian populations, we studied 4 alternative evolutionary scenarios. The first one, ignoring the sea level variation and LDD events. The second one, considering sea level variation and ignoring LDD events. The third one, ignoring sea level variation but considering LDD events. And the last one, the fourth one, the more complex one, considering both sea level variation and LDD events. Here, in this video, you can see a demographic simulation under the simplest scenario that ignores both sea level variation and LDD. Population gradually expands over time through the map until the map is fully colonized. In the second video, you can see a simulation of the most complex simulated scenario that considers both sea level variation and LDD. The colonization of the map is quite faster. Population starts to expand with more available land, and then population expands to that land that becomes available when the sea level reaches its minimum. Then it contracts when sea level rises and floods the region. For each scenario, we performed 300,000 simulations based on demographic and genetic parameters drawn from uniform distributions based on previous studies. Then, we computed the summary statistics from the observed and the simulated genetic data. We selected summary statistics related with the genetic diversity and with the genetic variation. In the end, we used a total of 22 summary statistics. Then, we selected the best fitting evolutionary scenario with approximate Bayesian computation to validate our selection. We evaluated if the applied method to perform the selection could correctly distinguish the summary statistics obtained from each scenario. Also, we evaluated the fitting of the observed data with the selected scenarios. In the final step, we estimated the demographic and the genetic parameters under the previously selected scenario. Regarding our results, our reconstructed phylogenetic tree shows a large admixture between and within populations. Here, you can see the phylogenetic tree colored by group, and here the phylogenetic tree colored by population. You cannot find any population isolation. These results are interesting, and we wonder which scenario may better explain this pattern. The best fitting evolutionary scenario considers both sea level variation induced by LGP and long distal dispersal events, as you can see from this table that shows the higher posterior probability to LGP and LDD. As I referred, we evaluated the power of the ABC approach to distinguish the summary statistics generated under each evolutionary scenario. The applied approach correctly assigned the summary statistics to the evolutionary scenario more than 80 out of 100 times. You can see in the table the scenario to which the summary statistics were designated. In bold, you can see the correct classifications. For example, for the summary statistics generated under the scenario that considers only LDD, the approach assigned it 89 times to the correct scenario, 5 wrong classifications as the scenario that considers both LDD and sea level variation induced by LGP, 2 wrong classifications as the scenario that only considers sea level variation, and 4 wrong classifications as the scenario that ignores both processes. Here, in this figure, you can see a graphical representation of the table. If all the summary statistics were correctly assigned, each column would only have one color. We also analyzed the fitting of the observed data with the selected scenario that considers LDD and sea level variation. This was performed with principal component analysis of the simulated data under the 4 alternative scenarios. As you can see, the projection of the observed data, represented by the cross, falls within the scenario that considers both sea level variation and LDD events, in red. Regarding the selected scenario, it involves continuous range changes induced by the sea level variation and migration through LDD events. Indeed, LDD events have already been proposed to have influenced the expansion of early modern humans through the world. The process is associated with lack of population differentiation and contributes to high levels of genetic diversity, patterns found in our observed data. Concerning the sea level variation, Recent studies have shown in other species that the land bridges may have not always generated high gene flow, particularly because the local environment influences the migration of the species. Here, our analyses show that the sea level variation is necessary to properly understand the genetic diversity and admixture of current Southeast Asian human populations. Possibly, the approach islands favor the sea crossings, and the land bridges contributed to gene flow by land between distant islands. Our estimated parameters agree with previous studies that suggest large populations expanding rapidly from Southeast Asia mainland towards Oceania 60,000 years ago. Here, the time of the beginning of the expansion and the population size at that moment. The growth rate and migration rate, carrying capacity, mutation rate, and long distal dispersal proportion. Even though mode, mean, and median not always match with previous estimations, our intervals always do. Overall, our results show that the sea level variation induced by the LGP and migration through long distal dispersal events explain the current genetic diversity and population admixture. Sea level variation should be considered to understand the expansion of early modern humans and the resulting genetic patterns. And also, the estimated parameters under the selected scenario support previous studies, meaning a rapid expansion of large populations from Southeast Asia towards Oceania. The most important message that I would like you to take home is that past environmental changes should be considered to properly explain current genetic patterns. Thank you all for your attention. I would like to thank the organizing committee. Importantly to the study, I must thank to Miguel Arenas, who supervised the study, and Marina for the help. These were the institutions that provided financial and technical support.